I'm honored to introduce Dr. Arboleda-Velasquez. Now, he's an associate professor of ophthalmology at Harvard Medical School and a principal investigator at the Shepard's Eye Research Institute at Mass Eye and Ear. Trained as both a physician and a scientist, Dr. Arboleda-Velasquez leads a translational research laboratory that integrates genetics, vascular biology, biochemistry, and preclinical therapeutic developments. Now, his work is distinguished by a dual focus on understanding the pathobiology of the small vessel disease, including CADASIL, and uncovering mechanisms of resilience to neurodegeneration, particularly in Alzheimer's disease. Now, Dr. Arboleda-Velasquez received his MD from the University of Antioquia in Colombia and a PhD in cell developmental biology from Harvard Medical School. He completed his postdoctoral fellowship in cell biology and vision research at the Shepard's Eye Research Institute, and his early work has illuminated how notch signaling regulates vascular mural cells in cerebral small vessel disease, including vascular dementia. He pioneered the use of vivo models to demonstrate the causal role of mural cell loss in diabetic retinopathy, advancing our understanding of vascular contributions to neurodegeneration and eye disease. More recently, Dr. Arboleda-Velasquez has become internationally recognized for his discoveries of genetic variants that confer protection against autosomal dominant Alzheimer's disease. Now, his laboratory plays a key role in contributing novel insights into the role of vascular protection and interactions in delaying the onset of dementia. Now, his team has developed antibodies and proteins to therapeutically mimic these proactive effects, work that is continually being advanced towards clinical application. Now, by bridging human genetics, vascular biology, and therapeutic innovations, Dr. Arboleda-Velasquez, his work aims to redefine how we understand and eventually prevent neurodegenerative and microvascular disease. Now, through collaborations across Colombia and the United States, his research is shaping the future of personalized medicine for both the brain and the eye. Let's give Dr. Velasquez a warm welcome. Thank you.
Thank you, Carrie, for that great introduction. And I first want to thank the organizers for giving me the chance of presenting today. Before I begin my own talk, I want to say that I was very inspired by everyone that I met earlier today. And in particular, I met this young woman here that said something quite remarkable. She said, you know, I work with CADASIL patients and I want to devote the rest of my life to working on CADASIL. And I actually want you guys to meet her. Her name is Megan. I said the same thing 27 years ago. And here I am still working on CADASIL. So I want to give, this is a cameo, so I want to give a chance to Megan to talk about the great work that she's doing.
Hello. So my name is Megan Caruso, and I'm a research project coordinator for the CADASIL Consortium site in Providence, Rhode Island. Over the last 3 years, I've had the privilege of working with over 100 patients and their family members with CADASIL and just learning so much about the disease. But like many of you, 3.5 years ago, I didn't know what CADASIL was. So I didn't really know what I was getting myself into, and I didn't realize how much this community and the people I've been able to work with and the families and the passion that everybody has for advancing this the knowledge of this research and this disease for the community would have on me. So I have been able to work with the CADASIL Consortium, which is the largest cohort of people with CADASIL and their family members in the world. We have over 560 people enrolled across 12 sites in the country. We're continuously advancing and expanding our research and just trying to understand CADASIL better. And this project has really made an impact on me, and I wish to continue furthering this research for the rest of my career. I'm planning on applying to medical school in the spring, and hopefully will become a physician and a scientist working with CADASIL patients and to advance the research and understanding of it for the entire medical community, because I want to echo everybody who has spoken before me that it's not well known, and it's not well enough known, and I've had the opportunity to go to the American Association of Neurology conference a couple years ago and speak to some neurologists about it, and while most people knew of it, they were like, oh, I've seen it once in my lifetime, but as Michael said, there's, 3 families within a, 10-mile radius of it. So it's extremely underdiagnosed and misdiagnosed, and anything I can do to help further the knowledge and the research, it's, I think I found purpose in life. So thank you so much for allowing me to talk.
Thank you, Megan. And again, I just thought it was very inspiring because when I was a medical student back in Colombia, I think I was 22, more or less, and I asked my mentor way back then, I was kind of like in a crisis in my life. I didn't know if I want to stay in medical school. I thought, maybe it wasn't for me. And he told me, well, how about research? And I was like, I like research, but all of you guys are working on Alzheimer's disease, and I'm really smart. By the time I finished my career, probably Alzheimer's disease will be like cured. Why don't you give me like a different project, something that nobody cares about, something that nobody's heard about? Give me that. And I was like, I do have a project like that. I have these families, and I think they have CADASIL, but nobody wants this project. And I was like, I want that one. Because I'm so smart. In like 20 years from now, 30 years, then, that's gonna be like where the future is. I wanna make a difference there. So he gave me that project. And much like Megan, I got super inspired by meeting all these families in Colombia, and then I ended up coming here to the United States. In Colombia, I got great medical training, and I ended up getting my degree, but I wanted to become a scientist, and again, I was very young. I was like, I'm gonna find the cure for CADASIL, right? That's kind of like the dream. I'm gonna help these people nobody cares about, And but for that, I was like, well, I need to become a scientist. And in Colombia, it's not that our PhD programs were bad. It's just that we had none. You couldn't get PhD there, So I came to the United States, and I ended up getting into the PhD program at Harvard Medical School, and I trained like a famous scientist there that had discovered notch. Have you heard about that? Like notch? That's notch, right? Notch rings a bell. But which one? Notch 1, 2, or 3? 3, right? That one rings a bell, right? Notch 3. There's the 1, 2, and 3. He discovered the original, the notch. Okay. And as luck has it, he was at Harvard, and I got the privilege of training with him on CADASIL. And I did my PhD on CADASIL, and I still work on it. Currently, I am funded by the National Institutes of Health. Yeah, they still fund research, Despite of all this, I have funded from NIH. Specific for CADASIL research. And I think actually I've been funded for CADASIL related research for the last 15 years, uninterrupted by the NIH, and we are very grateful for that. Now, one thing I learned this year And I mean, I knew it kind of, but I thought maybe that wasn't that, but it was just me. Communicating with people is very important. Like telling people about our research is very important. And we have to communicate in ways they understand. And I think that's something that our current president did really well, and the current administration did really well, is make everybody aware that people via taxes are the ones that pay for research, right? And we scientists think we know it all, and we talk to each other and we think we're super smart, and then we speak in terms that nobody understands, only us. The ones that know, the ones that go to Harvard. We are the ones that speak the language, right? And then we want money from the people that don't. Then you guys don't speak the language, but we want your money, and then we do research for ourselves. So I want to change that. That has been my mission for a long time, but this is something I obviously have done in the past, and I'm doing it today. And you're like, he speaks weird. Why do I understand him? He's supposed to be a scientist, and I actually get it. So that's part of it, because it's we need your money. We want you to keep paying taxes so that that goes to NIH, and then we get the funding so that we can do research. But I think it's the least that we can do is that you guys understand and you know what we're doing. So my approach to communicating science is not to dumb it down, is to actually speak it in a way that elevates knowledge so that we are all on the same page. So let me start by talking about I want to give you guys the chance to speak, not only Megan, but she can also participate. She has something to say. Why are you guys here? I suspect something. I think it's for what happens after me, like the music. That's it. I think everybody is here for the music, for the band. But in case you guys, some of you guys came from like far away. Why are you here? What do you want? Anyone? What did you hear I was going to talk about? Like, what's going on here? Or is it really about the music? Please be honest. A treatment. You guys want a treatment.
A cure.
Even better. A cure. I have a story about, have you guys heard about Cure Cadasil? Right, the association, right? I have a story about cures. Something had happened in my life, in my family, and I was like, I really want to do something for Cadasil families in the United States. So I create a Facebook group, and I call it, I was like, I want to be brave, I want to take things to the moon, I want to call it Cure Cadasil. Because we scientists are not supposed to talk about cures. That's too much. That's like, snake oil, right? We're not supposed to. I was like, I'm gonna call it Cure Cadasil, and then the group became popular, and then someone wrote to me. Her name was Barbara. Does that ring? What is Barbara? And she had like an accomplice. Her name was Anne McGuinness. And they were like, Joe, we want to start an association focused on Cadasil, but there is a problem. We want to call it Cure Cadasil. And you took that name. So we want that name. So you can't have to give it to us and not use it anymore. And I was like, okay. And Barbara, before Think Cadasil, she was one of the co-founders of Cure Cadasil that now became like this huge organization. So you came here thinking about a cure, and cures are important. So you want to hear about treatments, you want to hear about cures. Anyone else? Okay, that's good enough. Go ahead.
A friend of mine was diagnosed with the disease, and I'm a nurse, and I know nothing about it. I never heard of it before. So I came here to show support for my friend and learn about the disease. Like, what is it? Is it a dementia? Is it an Alzheimer's?
Thank you so much. And what's your name?
Marion.
Well, support is very important, and I'm really glad that you're here to support your friend. One thing that I want to talk about is, you're saying here is support. I'm supporting someone else that has. So how do we get to a treatment? How do we get to a cure? That requires a partnership. And I, put this together here. Let me see. If we can make this work. So this is what we want. We want cures. Let's go with that one. Why not? Is this possible? Yes, it's possible. What is it gonna take? It takes a village. Have you heard that expression? So in this village, I think, let's put myself here at the bottom. I am the scientist. I am the scientist, I'm here at the bottom. Now, we want the same thing, we want a cure. We cannot do it alone. You guys play a role here. What is the role that you play? What else do you think? Well, here is the patients, right? Patients with CADASIL. And we want the same thing, right? We are talking about the families. So let's put them here, the families. What else do we need to get to the cure, if this is the goal? What else is necessary?
Money.
Thank you. We need money. Where do we put the money? Let's put it here. We need money. How much?
A lot.
Okay, more. What else do we need?
Research. Awareness.
We need research, we need awareness, we need knowledge. So we need, the scientists, we do the research, but we do it with the patients. You guys are participating in clinical research. That's what Megan is doing, right? Like, let me tell you, let's say we have the cure. And we don't have the patients for the clinical trial to test it. So it will stay in my freezer, right? In the lab.
Yes.
There's actually a freezer in my lab that has things that could help, right? But unless we can test them, we cannot get to what we want. So we need the research, and let's put it here. And the research is clinical research, and I would say basic research, so we need both. What else do we need? We're missing one, but I think it's important. What was that?
Make it public.
Yeah, everybody's talking here about awareness, and I think that's very important because, it kind of like, if people are not aware, Megan doesn't get her patients, we don't get the funding, people that have the disease are not diagnosed, there's no way to know, so it's awareness. We need to be aware of it. And I'm not surprised that you guys are not mentioning this one because they're kind of like in the doghouse right now. It's like biotech and pharma. Nobody likes them now, right? We need them. They play a role. So we actually, why do you think we need them?
To develop.
What?
To develop.
They got resources.
They get resources,
Influence.
influence, they have the know-how,
Money. They have money.
they have money, they get, so it's biotech. Thank you for saying that, because the money a lot of the times comes from the government. We talk about, actually let's mention that, like money comes from government. In this case Trump, okay? So it also comes from investors, right? This is good because now you can tell if a scientist can spell or not. We need investors. Some people, they are willing to support CADASIL research, but they want to make money in the process. Is that the end of the world? I don't know, but it's one way to do it that has been used for other things. So there's biotech, and the moment that you get to connect these things, you make the cure possible. And I actually want to say something. I actually think we are going to find a cure for cancer. I think we will. Actually, I'm positive we'll have a cure for cancer. How positive I am. 100%. And you're like, okay, he's the snake oil guy. How do we pay for this? So I am because there is the then. Now, I think you guys probably agree with me because you know that research science, it makes progress. So the key question is when, right? Because you know I'm right. The thing is it going to be next year? Is it going to be 5 years? And is it going to be 10 years? And how long is it going to be? What is it going to take? That is what makes a difference for us, is the when and the how. And that's what separates the salesman from the person that actually delivers. Okay, it's the when. Now, just when do you since you guys seem to want it, when do you want it? Now. Okay. That's kind of fast. But guess what? The now, as soon as possible, sooner than possible. We want it now. Let me tell you when I wanted it. I wanted it when some of my patients were alive, because I have patients that I met almost 30 years ago, and no, they are not around anymore. That's when I want it. So guess what? I'm more ambitious than you guys. I don't want it now. I want it 30, 20, 10 years. I want it in the past. I actually think we're late for calcium. Calcium was discovered in 1996. So what's next year? 2026, right? What's that anniversary? The Pearl anniversary. 30 years. We're late. We're late. And there are barriers. There are barriers that separate. I'm putting this thing here. There are big barriers that separate cures from us. There are big barriers in the field. I mean, I have tried to overcome many of them. So I just wanted to put this in context. Also mention to you, I'm required to do this. I am an academic professor. Unfortunately, I work at Harvard, That used to be a good thing. Not too long ago. I have my lab there, and we're doing great research there. But I'm also required, because I'm an academic, to do what we call a disclosure. At some point, we academics, you guys, you have your needs, and I know you want a cure and all that, but guess what? Sometimes we, the academics, have to make the discoveries, and we have to raise the money, and we also have to start companies to then bring that to people. And let me tell you why. Because 30 years into it, nobody cares. I care. Megan cares. Nobody cares. I do care. Yeah, I have to start a company. It's part of my disclosure. It's called X1 Biotech. I have to tell you that. Where was I getting from? Okay, that's just for what it is. It's called a disclosure. You know how companies look at disclosures? They love it. It's advertising. But from an ethics perspective, it's kind of like, so that you know that when I speak, there is something in the back of my head. That's kind of like the way disclosures work. So yeah, I'm involved with this company. I had to start it because if I didn't, things would actually take longer. Now, what I'm going to keep talking to you about today is regarding my research. I cannot really speak on behalf of the company. I just have to disclose. Any questions about this, or can we move to the next chapter? Okay. You never heard the scientists talking like that, right? I don't think so, okay. You're not supposed to talk like this. You're not supposed to talk chop. But, I think the current times called for. Now, I want to tell you about two things that I think are very important for you. One, I think it's important to talk about Cadacil. You had a question again about like what is it? My friend has it. What's happening here? We all know about Cadacil. It's either dementia, it's a small vessel disease. What is it? The way I think something that everybody here has in common is like this gene called NOSH3. It's a gene. We all have it, but some people have like different flavors of it. In some of them the spelling is right in the whole sequence of the gene, and in some others there is like a small spelling mistake. So when the gene is read, the protein that comes out of it is weird. That's a gene. A gene makes a protein. And in the case of Cadacil, yeah, there is a problem. It does look weird, trust me. Sometimes it's not even there, but it's hard to see. So the moment that you have this problem in the gene called NOSH3 and the protein is weird, there is a problem in the arteries of the brain. Why do we need arteries in the brain? Because we need oxygen and we need food in our brain. Most of the food and energy that we take is for the brain. Even, it takes it all. So the arteries are the ones that bring that nutrients and that oxygen to the brain. And in Cadacil, when there's a problem in NOSH3, the vessels degenerate. The vessels burst. The vessels occlude. In other words, they're messed up. Okay? If anyone here has a hose in their house and the garden and something like that, the hose is the artery. And that's the one that brings the water to the plants. There is no difference. Now, who likes to work with a hose that is messed up? It's broken. It's leaky. When the guys take you to these MRIs all the time, I've done an MRI once because my wife offered me $50 to do it for a study. And I was in there, I was like, I'm gonna do something really bad with those $50. Like, holy nice kid, this is horrible. I don't know how you guys do it. It's a terrible experience. I learned I was claustrophobic when I took an MRI. And I don't even remember what I did with those $50, but it was bad. So when they take you to the MRI and they look at your brain, what they see is the evidence of those leaky vessels. And then they tell you about those white spots, like the leukoencephalopathy. Oh, we're so smart. Leukoariosis. Microinfarcts. Yeah, I know them all. There is, in any case, so those are vessels bursting, okay? And that's what happens when the protein is messed up because you have a NOSH3 problem. Now, the confusion comes, the moment that the brain doesn't get the nutrients it requires, neurons die. The neurons, everybody knows, those are the ones that we use to think, okay? To remember, to move and to plan things. Finding something in your wallet and in your purse seems easy. Ask someone that has Cadacil to do it. They can do it. It just takes forever. It's called, executive function. Have you heard about, you'll get about Megan, right? Executive function. That is executive function. That takes planning. You first have to open the zipper, go into the one pocket, find the little baggie, and look for it. That takes planning. It's called executive function. You actually have to remember it was there to begin with. So it's a combination of things. But the moment the neurons die, and we don't have them, because the vessels burst, you start losing cognitive function. You lose your memory, you lose your ability to plan, to be independent, you lose mobility, and maybe you can still handle yourself, you still have a job, and you are independent. If you are independent, you don't have dementia. You were talking about, is this a dementia? At some point, so many arteries in the brain are so messed up that so many neurons die. You're not independent anymore. And the moment you're not independent, that means you have dementia. So yes, to answer your question, CADASIL can lead to dementia. It's hereditary because it's caused by mutations in a gene, in this case called NOSH3. And a lot of the times, when the arteries burst, it could be that progressive and people just get worse over time. Sometimes it's sudden. I was fine this morning. I think, was it Michael telling us? And then all of a sudden I couldn't speak. I couldn't move. That's a stroke, right? That's like a very sudden kind of like loss of cognitive ability. So that's kind of like the difference. Is it progressive or is it sudden? So this is the background that I wanted to give you about like the disease. Any questions about this or are we good? How many of these we have? I'm going through them very quickly. Okay. They gave me a lot of colors, so I feel like I have to use them. Otherwise, let's change, let's go with orange.
Pick a dark color so we can see it better.
Oh, that's good. Black.
Yep.
Okay, thank you for the suggestion. And please feel free to interrupt me or ask any question. I am making an effort to speak in ways that you understand, but there might be something that I completely missed. So let me know and interrupt me, or if anything I can do better, or if it's a word I didn't explain well. So what are the two studies that I'm doing right now? One, I call it my moonshot, and this is the one that's being paid. People say about the NIH that they pay like boring research and that's not true. It's not true. They are paying right now with your money, with your tax money, they are paying for my research, and it's a moonshot. What I told them was the following. Eventually, for personal reasons, I ended up working on Alzheimer's. I know, boo-hoo. And I made a big break. You guys were talking about cures, right? How many cures do you think I found for Alzheimer's? 0, 1. Nobody said 2. I found 2. Yeah, look it up. Look it up if you don't believe me. This one, look this one up. It's called ApoE. Look it in your phone. Christ Church. Look this up on your phone. See what you find. I published that in 2019, and it was everywhere. Front page of the New York Times, and every other outlet above. Okay? That was big news because it was this woman that was supposed to get Alzheimer's in her 40s, and she got it in her 70s. And we identified the gene that protected her against Alzheimer's. That was discovered in my lab. I really apologize. That's besides the point. Then I was like, we can find these genes that protect people against Alzheimer's. Can we find more genes that protect? And we are developing treatments based on this. Why is it a cure? Because if somebody that is supposed to get Alzheimer's in their 40s, get it in their 70s, most people with Alzheimer's get Alzheimer's in their 80s, that lasts 30 years. 110, right? That's a cure. Okay? Then we found another one, and we published that again. That was also in CNN, and people were talking about that. It's called reeling colbus. We found another gene that protects people against Alzheimer's, reeling colbus. It was a collaboration between Colombia and Boston. Trump is fighting with the Colombian president. Trump is the smart one. Trust me. The Colombian president, not good. Okay, so reeling colbus. This one also protected people against Alzheimer's for 30 years. So at some point, I was sitting in my lab. Again, that we are developing treatments based on that, based on this. Like, it's a big story. Look it up. And at some point, I sat down and I was like, what happened with that promise I made that I was going to devote the rest of my life to work on CADASIL? What am I doing for the CADASIL patients? Can we find people that are naturally protected? Yes, I'm looking at you. Are naturally protected against CADASIL. And I'm sure we've been talking about that. And again, I mean, I don't want to put you on the spot, but you kind of like spoke earlier about your mom and how, you know, she unfortunately passed away at age 45, right? And you're here talking at age 47. So there is variability. There is variability in the clinical presentation of CADASIL and what we call the sophisticated terms like the age at onset. When did it start? And sometimes the variation can be huge. We're talking about that earlier. So could it be that some people have natural protection? And I was like, I want to fulfill my promise to CADASIL patients. And I proposed that to NIH. I was like, I was the guy that found Apoy Christchurch, and I was the guy that found reeling colbus. Can you give me money to find the protective genes against CADASIL? So in this case, it would be like the CADASIL protective gene. So the protective, not the one, not the NOSH3. How should we call it? The anti-NOSH3, anti-CADASIL gene, the one that protects against CADASIL, the nemesis of CADASIL, And they say, oh sure, that sounds like a good idea. If you can do it for Alzheimer's twice, that seems like a tough nut to crack. Maybe you can do it for CADASIL. And we're funded to do that. And it's a study, we study people, and some of them start really early, some of them start really late, and we study the genes, and hopefully you will see sometime later down the road a paper that talks about a protective gene against CADASIL, and I wanted you guys to know because you kind of like are paying for it via your taxes. So this is being supported by the NIH. It's a great collaboration with Dr. Yakel Quiroz. She was at Mass General, now she's at Boston University. And just because of the structure of the genetics, the work is being done with families in Colombia. We have what we call a genetic isolate. I'm from a region of Colombia where 5 million people came out of 8 families. We were all very Catholic. That's just kind of like, just have children and procreate, okay? Mandated by God, okay? So you just give it time and God's duty and you make 8 million people. So the genetics is such that you can really make very sophisticated genetic analysis. By talking to people here, I learned something, which is that maybe we should do that via collaboration and include patients in the United States, maybe for validation. Megan, need your number. Okay, so this is where we, at these events, she meets people, I meet collaborators. This is not just the food and the music, okay? So this study is funded by NIH, you can look it up. It's going really well. I actually, since you kind of like came all the way here, you paid for it, yeah, I think we might have found one. We might have found one. We might have found one. Again, I'm a very rigorous scientist. Of course, I need to tell you what we call a candidate gene. It's a candidate. So you're running for president, but you're still running for. So you're not the president yet. Does that make sense? But it's been nominated. We actually call it a nomination process. You run for it against the other genes, and you kind of pick up the best. Okay? So you run for it, and you won the nomination, and we have a candidate gene that we think protects against CADASI. Okay? So, you guys will be hearing about it hopefully soon. The NIH the candidate is so good, we're going to be reporting it to NIH already. We're meeting with NIH early next year. They want to hear about it. They're very excited. Actually, the institute that pays for this, they don't care about Alzheimer's. It's called the National Institute of Neurological Disorders and Stroke. So they are the ones supporting this research. They support the NIH. It is supporting our research. Any questions about this? It's a very important. Oh, yeah.
When you say that this research will protect against cancer, what about those in the later stages of cancer?
Okay, so for those, let me tell you what it happens. Like, let's say when we find a gene that protects, that is kind of like what we call an insight. It's kind of like, oh, this gene protects. Can we make a drug that does the same? And hopefully the drug can help people at all the stages. So this is early research, and it's kind of like risky, and it's a little bit removed. Even if I publish a big story about the protective gene, that is still some time away from like an actual drug. The next story that I'm going to tell you is more proximal to like this, more tangible. It's closer to like a drug. I do wanna, and let's get to that right now, because again, this is something that you should know because you're paying for it, but it's not the drug you want because you cannot say it now. This one is not now. And again, it's so early that I don't even know if it will help people that already have it. That would be irresponsible of me to say because I don't know. It's early stage. So now there is another type of research that, again, and it's inspired by what happened with the Alzheimer's story. At some point, again, I reflected and I was like, why have I been so creative and have made such great groundbreaking research in Alzheimer's, but I have not done that for CADASI? No. She was saying it's because it's more complicated. Alzheimer's is pretty complicated, and it still brought that one. What do you think that was? What do you think I haven't really, what do you think went through my mind when I thought that? What do you think sometimes we work so hard on something and we just don't get it? We just can't do it, but we are successful on other things. Huh? You got it, too close. I knew too much. Right in front of me, I couldn't see it. That's what happened. I was like, I want to give myself the chance of being as creative as I have been for other things, for CADASI, which is the thing that I promised myself I was going to do in my life so many years ago. And I started to see what was too close to me that I couldn't even see it. And let me tell you what that was. I had worked on another gene. Let me tell you about it. It's called RANX1. It's also a gene. I started to work on that 2015 with a collaborator of mine, Dr. Leo Kim, and we discovered that this gene called RANX1 is called a transcription factor. A transcription factor is very, it's a sophisticated name for something very simple, which is you have the genes, but Not all the genes make all the proteins at the same time. Transcription factors regulate that. So it's a transcription factor, but that's just a sophisticated name to say what it does. Now, this gene, we found that it was very important for another disease that affects people with diabetes. People with diabetes, let me see. You guys, you're very smart, you're gonna see where I'm going with this one. People with diabetes, they go blind. You know why they go blind? Because the arteries in their eyes burst. They leak. They bleed. And we found that RANX1 was very important. This is a representation of the eye. It's called the retina. These are the arteries. And the Eye Institute, again, I'm sorry, they were the ones that gave me a job. The neurologists weren't interested. But you know, these are the arteries, they burst, they leak. That actually also happens in the brain. People with diabetes, high blood sugar, they also get dementia, and it's because the same thing happens in the brain. And we published that this gene, when it gets activated, promotes this vessel vascular arterial pathology that affects the eye and affects the brain in diabetic patients. And we published that in 2017. That was very exciting news that made it to the cover of the journal Diabetes from the American Diabetes Association. Big news. Then this result was later validated by other people. External validation is very important in research. You don't want to be the only one doing something and finding this, one thing. So it got a lot of external validation. Then we found that this was also very important in any type of arteriopathies. Like that can also happen. Is this being recorded? This. Okay, you get the idea. Everything that affects the arteries. We published many other papers on this. 2017, 2018. We published a big paper just last year in 2024 with another company. This result has been very thoroughly validated, even in the lung. This is a thing. RANX1 causes arterial disease that, in many conditions, and the moment we use a RANX1 inhibitor, there is this drug that inhibits RANX1, and the moment that we give this drug to models, we can do that in cells, we can do that in mice, we can do that in rabbits. We have done a lot of animal testing on this. Sometimes, I'm a pet owner, I care about animals, but we think it's important to do some studies in animals, and we do it in a way that we think will, mitigate pain and suffering for the animals. But yeah, we do animal research. It's also important. Now that's kind of been phased out, but yeah, I did animal research, and we found that the moment we gave this RANX1 inhibitor drug to these animals, the arteries were preserved, the vessels did not leak, the vessels did not burst. And that brings us to, that has been thoroughly validated. We published many papers. And then all of a sudden I was like, again, I want to be very creative for CADASIL. I want to find something for CADASIL. I want a therapy. And I don't see it because it's too close. How close? Like real close. I've published on it in 2017, and then in 18, and then in 19, and I got grants for that, and it was so close I did not see what it was right in front of my face, that maybe the same drug that was good for the diabetic mice and the rabbits and all of these things could help Cadasil. And that was when the light shone upon me, and I was like, aha, I want to test this hypothesis. I want to see if this same drug that I'm going to give you the name of it. Okay? It's called Raw 2479. And I studied the name so that I would get it right. But I think I got it wrong. And it's hard to remember names. That's why I want to call it something just here. We're going to call it Rory. Why Rory? Because it's the raw drug that is a Ranix 1 inhibitor. She's a Rory. It's a cool name, right? Let's call it Rory. So Rory is this drug that is a Ranix 1 inhibitor. And we actually picked this drug for our studies for a reason. And the reason was that this drug has actually been in humans before. So this very same drug had been studied in humans back in 1995. It was advancing clinical trials for people that, they say they had HIV. These people had AIDS. If it was 1995, you didn't have HIV. That's like they are trying to whitewash something. They have AIDS. 1995 people were dying with this thing. And they gave them this drug because they thought it could help with this situation that they had. They didn't know it was a Ranix 1 inhibitor. They thought it will actually do something to the virus, okay? And as it turns out, for AIDS, it didn't work. But it was found to be relatively safe. Nobody died. The only problem the patients had was a rash, like a skin rash, sometimes. Not very common, but it was there and it was reported. And the reason for Roche, that was the company that I had at the time, not to pursue was the lack of efficacy. The patients just did not get better, and then better drugs got into the market, and everybody forgot about this drug, and that was it. But as it turns out, later on, someone at the NIH themselves, they found out that this drug, Rory, was indeed a Ranix 1 inhibitor. And we decided that if we focus on Rory, we could skip a lot of steps that normally have to be taken for drugs to prove safety and a lot of things, and things could move faster. Because again, you want things now. I want things now. I already feel like we're late. So this exercise is, you can call it, and you do research on it, it's called a repurposing exercise. It's called a repurpose. When you repurpose a drug that already has been in a clinical trial, that has been through all sorts of animal studies, that has been already in a phase 1, that has been in a phase 2. This was actually a phase 2 trial. And people received the drug for like 3 months. So it's already kind of like a lot of knowledge about the drug. The only thing that is new here is that we think it could work for like the diabetes thing. So what we did is, I actually remember now, it's raw 24, 7429. And I'm going to keep putting numbers until I get it. But the bottom line is that Rory, you can look it up. That drug is commercially available. People use it for experimentation. Please do not take this thing. We don't want like a chloroquine situation here, like people go in and take things that they sell. That's for like animals studies and things like that, or for like cells. Things that if you can buy it online, it's not for people. Okay? Does that make sense? Like, to make it very simple, it's not, if you can just go ahead and buy it, it's not for you to take. Like, because it's the way it is produced. Things that we take are produced in a way that makes it safer, so please don't. But, this thing is out there, people can buy it and do experiments with it, but don't experiment on yourselves, okay? So this is a repurpose exercise, and then I was like, okay, we know Rory works in diabetes, we know Rory protects vessels, can Rory work on CADASIL? And in my PhD, I have made animal models, so we have different animal models in my lab, so we made, in CADASIL, I'm sure you will be familiar with these things. It's like a letter that is usually a C, followed by a number, which is the position in notch 3, and then followed by an R. Or it's an R and it changes to a C, or it's always a cysteine, right? This is an abbreviation for an amino acid called a cysteine. And it's always, and look at your reports if you have been diagnosed with this, there is always a C somewhere involved, and another letter, usually an I. So I had made a mouse inspired by one of the Colombian families that has this CADASIL mutation. I had made another mouse. This is an R, followed by a number, followed by a C. That's again another type of CADASIL mutations. And another one is what we call the notch 3, kind of like knockout, like a null. And there is a third model I cannot tell you about, but it's kind of like boring. But we have tested in like 4 models already, of which I can tell you about now. And now, most scientists, because of what we do, is kind of like proof of principle. You just want to know if maybe it works. Sometimes we kind of like, you can do experiments in a way that help the experiment. Does that make sense? If somebody likes cooking, it's called the helper. You put something in there so that it looks like there is more food, or it tastes better for the kids so that they will actually eat something. And that's why instead of the chicken, you make nuggets, okay? So we do experiments, and instead of chicken, we make a nugget. So, but in this case, I didn't want, and sometimes that translates in, we do experiments in animals when they are not sick, and then treat them for like a very long time, so that they never get the disease or something. That experiment is easy to work in a way. I did not do that. Somebody was talking about this. What was your name? Yeah, I'm talking to you. The one that wanted, like, if you work in people that already have it. What was your name?
Tasca.
Tasca?
D.
Tasda. Tasda was like, can we do this? Can we use this for people that already have CADASIL? Yeah, I used those mice. I used mice that were sick. How sick? Very sick. The vessels were messed up. The arteries had bursted. They were in trouble. I used those. The ones that, how about we just don't forget about them? It's going to be really hard. No, I used those. So I used those sick animals, and I treated them. I know how to spell sick. Come on. Okay, so I treated these sick animals. They were 6 months old. By that time, those animals were sick. Okay? And I treated them for a month. One month on. Now, before I tell you the results, this happens to the Alzheimer's people. They actually found a treatment for Alzheimer's. Biogen did that. It was called aducanumab. And that happened like 3 or 4 years ago. Did you hear about the story? Who wanted that drug? Nobody. Who could pay for that drug? Apparently not even the United States could pay for it. They made the math. It's like $50,000 times everyone we have with Alzheimer's. Oops.
Why was it so expensive?
What was that?
Why is it so much money?
It was too expensive. That's what the company wanted to make. They were, well, we invested money, we want to make it very expensive. And not even the United States, the wealthiest country in the world, could buy. And there was something else that was terrible about that drug. People had to go somewhere to get an infusion, not different than people have to sit for chemo. We've seen it in movies, right? They plug themselves to the machine for like an hour or two. Yeah, those. Those are the infusions. And you have to do that every 3 weeks or every month. How easy is that to do with CADASI people? Taking them somewhere for an infusion every month? Not at all. So, this drug, Rory, is a small molecule. What do we mean by that? It's a pill. You can pop it in the comfort of your home. That's how the people with AIDS took Rory way back then, the pill. Okay? And this drug, this is how the drug is administered. It's a small molecule, it's a pill. So, but that solves the thing about how people will take it. How expensive it's gonna be, well, we have to see about that. That depends on you guys. The communities can lower prices by, I don't know, they call it heckling, sabotaging, protesting. I mean, who am I to give you ideas, I told you my disclosures. So, again, but again, the idea is that this is a small molecule. So what happened was, I don't know, the impossible happened when we get this thing. My student came back with the first data, and I was like, it's not possible. It's not possible because what she showed me was this, and it's very simple to understand. People is like, yeah, I'm not going to tell you because you're not going to understand. Every time you hear anyone telling you that, they are lying to you. It's not that you cannot understand, it's just that they don't know how to explain. So send them back to school, where they come from. I don't know. So in CADASIL, let's say the CADASIL mice, it's very simple. Like let's say this is the retina. We look at it this way, and we have different types of arteries, or vessels, if you will. We have the arteries, and we have the veins. Believe it or not, the arteries look thinner because they have muscle. You heard about smooth muscle, right? Smooth muscle in calcium goes away. So because there is a tone, they look like tense and hard. Instead, these ones, the veins, they don't have that much muscle, they look fluffy. So they widen, they like fluff out, okay? So by just looking at the retina or the brain, you can actually tell if it's normal or if it's CADASIL, because in the normal you can see thin, wide, they alternate. It's very easy. Anyone can tell the difference because the arteries and the veins alternate. And then when this is normal, if you will. In CADASIL, you guys are really smart. How will all look like? Will they look thin or they look like dilated, fluffy, no muscle? Fluffy. So they all look really fluffy. So they all look like veins. You don't have to be a genius or go to Harvard or Stanford or Columbia, or they all got in trouble this year. So you don't have to go there. They all look fluffy. They have CADASIL. It's leaky. So that was the expectation. After 1 month, they all looked like that. So it's like, hey, no thank you. It's probably not true. I think we first did it in this one. I was like, go back and repeat it. Another cohort of animals got the same. Then they did it in this one, same. Then they did it in this one, same. Then they did it in this one, same. And at some point I was like, okay, enough. I'm gonna start talking about it. because this is maybe potentially true. It's called replicating in different models, yeah?
So these three different, those are all mice, these are all different mutations, but they all make caddis.
Yes.
And
You
mice-
probably here have many, we all have, a lot of people here have cadassil. It's unlikely they have the same mutation. They probably have a letter followed by a different number followed by a different letter type of mutation. Yeah?
And the animals you tested on have caddis?
Yeah, they have something very similar
So
to
it- Okay.
Yeah, they have something very similar to calcium because the vessels that they generate, the arteries burst, they look weird. They look, yeah, it's like what we call a calcium model. We don't,
And the animals that you tested have the RUNX1 gene also?
they have the RANX1 gene and it goes up. Very good. So what we discovered, but what I want you to know is, yes, sometimes people like to talk about thinking outside the box, breaking barriers, doing things differently, and that sounds great, and nobody does it. In this case, I did it. So guess what? Yeah, I admit it. I first tested the drug and then did the biology. It's called thinking outside the box. It's called doing things differently. It's called let's just go ahead and do it. And when you read the scientific paper, it's probably not going to tell the story like that. I still need the orthodox academics to approve it and all that. That's in the process of it all, that's happening, otherwise it's not going to work. But the bottom line is, yeah, we tested it, and sure enough, in this case, RANX1 is up. In this case, it goes down with the drug. I think, we had a plan with Kelly. She was going to stand up, but it was, how long? Like almost 5 minutes. Okay, good. No, we made it. We made it. So Rory seems to work. The paper is in progress of getting published. So very excited about it. I just felt that it was important for me to share the great news with the community. The information, in a way, is public because we had put, as part of my academic work, we have to. Have you heard about like patents? You have to file a patent so that then people can invest and then a drug can be developed. So yeah, we file a patent and the patent issued. So this discovery, in a way, there is a patent and it issued. So the patent office declared that it was valid and that we own the rights, and that's important for my institution so that then companies can invest and things like that. So the patent issued. And again, it's like, how come that takes forever? Yeah, what I told you that happened in 2017, that's when we filed for the first time. Okay, so, and
So
eventually.
if you repurpose the drug, it has to have a new patent, even though it already had a patent?
Yes, it's called a method of use. Believe it or not, those are pretty useful. I mean, now, what we need right now is, where are we at? Yeah, the paper is going to publish. We formed the company some time ago. The company now focuses on cadassil. This Rory thing can work for diabetes, can work for cadassil, can work for many things. Our hope is to kind of like pursue the development of the drug, and the company is working on that. The hope is that when this has to go to the FDA, and the idea is that hopefully they will allow us to move to the clinic sooner, and it will still need to be tested. It's not like just because we test it in a mouse it works, not because it works in this is kind of like the part of the reality check, not because it worked it was safe in people with AIDS, it's going to be safe in people with cadassil. We still have to kind of like get FDA approval, we still have to do clinical trials, but I think it's going to be faster. That is the hope. And because you guys came here, this is the first time anyone has ever heard about this outside of my lab. So thank you for coming. I hope this night was worth it.